Welcome, everyone. I'm Craig Thompson, President and CEO of Memorial Sloan Kettering, and I'm pleased to welcome you here to the Paul Marks Prize Symposium. Our first recipient for this year is Simon Bolton, and to introduce him, I've asked John Petrini to come up and give us an introduction of Simon's career and his accomplishments. John.
Right. Well, I'm pleased to have this responsibility to introduce Simon and to tell you a little bit about his outstanding career. I've known Simon for a long time, since he was a graduate student with Steve Jackson, in whose lab he described the yeast version of the Ku heterodimer, which is important for non-homologous end joining in all organisms. But Simon also revealed its role in telomere maintenance and telomere biology. And I was kind of going through Simon's CV, and I, he's a friend of mine, and I really, I feel happy for him. But then you look at the CV, and there's this other part he's so young and he's done so much. It's kind of I'm kind of depressed right now, actually. I'll overcome that though, Simon. So he left Steve's lab after really, I should say the Ku work had a very big impact on the field, and about that was about the time where it was clear that the non-homologous end joining machinery was involved in antigen receptor gene assembly, and the detailed view of how Ku worked that yeast provided was really had a big impact very broadly in the field. And this is during his 3-year time as a graduate student, which is, I think, pretty impressive. So he then went to work with Nick Dyson and Mark Vidal, where he did something I think was really sort of forward-looking. He switched to organisms to use C. elegans and took advantage of the great genetic facility in that organism, and took the bold move of using it also as a place from which to do biochemistry. And that at the time was kind of, I think he were the first person to really do that on a big scale. And Simon used to have this slide where he showed there was a big vat of worms, and next to it was a slightly smaller vat of bacteria that grew and funneled into the worms. And we were all kind of in awe of this for a while, and we still are. But I mean, you don't show that slide anymore. So anyway, so with Mark Vidal, he really did some very interesting pathway description and pathway analysis. And it's here where Simon really began to get into sort of higher throughput, larger scale scientific analysis, and he did some beautiful screens that led him to later on the identification of a helicase called RTEL1, which is sort of an analog of the BLM helicase in humans that cause the deficiency of which causes Bloom syndrome. He really got a lot of information out of RTEL1 that is critically important. He told us about its role in mitotic cells as an anti-recombinase, also its role in meiotic recombination, in limiting the placement and number of crossovers. And more recently, I'm happy to say, in collaboration with my lab, he and I have also described the role of this protein in a rare form of dyskeratosis congenita in humans. So this arc of this work, it's, you remember Simon's very young, and so we still have to talk about his trajectory and so on, remains a positive and very steeply upward. And it reflects his ability to integrate a lot of very creative science and a very reasoned and sound interpretation of his data, and also just a huge amount of energy. And so I really admire him for that, and all of us in the field do. There's a lot I could tell you more about this beautiful iron-sulfur cluster story. Suffice it to say, there's some really great science here. But before I sit down, I want to also point out that Simon's very highly regarded and is a friend to many of us in the field, and this is because of the very positive and open spirit he brings to scientific interaction. And I think that really is one of the. I think about his data, but I also think about a lot about what a good scientific citizen Simon is, and it's really great that he's here as a recipient of this year's prize. Now, clearly, this is the most important prize that you'll ever win. I don't think that you've had one so far, but he has, of course, been highly decorated in from other places. In 2012, he was elected as a fellow of the Academy of Medical Sciences. He's in 2011, the Royal Society Francis Crick Prize was given to him, the Emble Gold Medal, and so on. So today we welcome him here to receive the Paul Marks Prize for 2013. Simon, it's a pleasure to have you.
Thank you, John. Can everybody hear me? Is this on? Yeah. Thank you, John. I didn't know John was going to introduce me, and the scary thing is he's already introduced the vast majority of my talk. But before I get to my talk, I wanted to take this as an opportunity to thank quite a number of people. And I think this is historically what the prize winners of this incredible award have done. First, it's a great pleasure to meet Paul Marks in person. I've heard a lot about you, and obviously I know a huge amount about your work. So it's great to meet you in person, and thank you for this prize. Of course, also thanks to Sloan Kettering as well for supporting this, and of course the selection committee, which in their insanity decided to give it to me after a number of years of persistence. But anyway, I'm very grateful for that. Also, congratulations to the other winners. I think we'd all agree it's quite a humbling experience looking through the past group of winners and what they've achieved and what they're still achieving. So there's no pressure on us, I don't think. So the other people, of course, I would like to thank are a special mention for Steve West. He's been a mentor and a friend and a great colleague to me, and he was the one who actually nominated me for this prize and the other prizes that I've had in the past. And he's been very influential on my career, so thank you very much, Steve. Also Thomas and Lindahl and Richard Treisman, who are the former director of Clare Hall and the director of the London Research Institute, who gave me the opportunity to establish my lab there, and they've been continually supportive during that time, so thank you to them. We have a great environment at Clare Hall and at London Research Institute that fosters fabulous scientific interactions, and it's all about the colleagues and the environment for me, and so a big thank you to them. We also have a very nice interaction with Sloan Kettering that goes on many years, and so it's a real privilege to come back here and see many familiar faces, which is great. So I thank my colleagues at Clare Hall. Of course, a lot of my work is not me, it's the people who I work with. I've had a fabulous group of students and postdocs in my lab over the years. They bring a huge amount of energy and different perspective, which is so important in science, so a big thanks to them. But most importantly, my family. I have 2 little kids, and they would have to be sedated if I brought them here. They would run riot. So it's really brave that you brought your family. I think it's great. But anyway, I have a son, Ronan, and a daughter, Iona, and my wife, Julia, and they kind of give me a huge amount of support and a perspective on science, which is very healthy, I think. So, to the science. As John mentioned, so I've actually worked on genome stability mechanisms for 20 years now, so I'm not that young, John. So what I'm sure many of you will appreciate is that the genome and DNA itself are actually intrinsically pretty unstable. The DNA is constantly under attack from both exogenous and endogenous sources. It's quite a frightening prospect, actually. But of course, we also exploit the DNA damage in cancer treatment and radio and chemotherapy. But the very act of metabolism, for example, produces free radicals that react with DNA, cause potential mutagenic changes that, if left unrepaired, will lead to transformation and potentially cancer. So, as I'm sure many of you are aware, the cell combats this array of changes using very specialized machinery that repair specific types of DNA damage in the cell. And my career has really focused on one type of DNA damage, which is the double-strand break. And you could argue that this is probably the most dangerous lesion in the cell because, of course, if you don't repair this, the genome becomes essentially split in two or into multiple pieces if you have multiple breaks. And also the way you repair these breaks, if you put different ends together, this of course can drive translocation, rearrangements, gene amplification events, which of course are the driving force behind cancer. So there are two major pathways of repair. There are others, but for simplicity's sake, the two that I will mention, John's already mentioned one of them is non-homologous end joining. Very simply, this is a gluing mechanism that essentially sticks the ends back together independent of sequence. That in itself is a problem because obviously we have breaks from different parts of the genome. They can be put back together, and of course you have a translocation. And recombination, which I've worked mainly on in the last 10 years or so, is essentially a photocopying mechanism that uses an intact sister chromatid or a homologous chromosome as a template for repair. So these are really the major questions that my lab has been addressing over the last 10 years or so, which is how you choose the different types of repair pathway, and this is very much governed by what position in the cell cycle you're in. As you'll hear in a minute, double-strand break repair is absolutely essential for meiosis, for generating gametes for sexual reproduction. None of us would be here without it. And so this is a major component of the work that we do. And also in normal dividing cells, we have major problems at replication forks. They're collapsing all the time, making breaks. And you need double-strand break repair to basically complete replication. And this is mentioned here. And I'll also mention some of the work that we've done on telomeres. So these are really just to break this down into 3 subcategories. So we have the spontaneous class of double-strand breaks. These, of course, are the ones that we exploit in radiotherapy, ionizing radiation. But as I've mentioned, a major source of double-strand breaks is a collapse replication forks, and that really is the challenge that we have. And almost certainly, it's the inappropriate or misrepair of these events during DNA replication that are the prelude to translocations. But double-strand breaks aren't all bad. I've mentioned meiosis. The programmed double-strand breaks are absolutely essential for generating physical connections between homologous chromosomes, and that helps them to segregate correctly in the first meiotic division. And so without this, you would not be able to generate gametes for reproduction. Furthermore, we wouldn't have an immune system if it wasn't for programmed double-strand breaks. So VDJ recombination, class switch recombination, which are important for the establishment of an immune repertoire, are all initiated by programmed double-strand break events. So we need them. So double-strand breaks aren't always bad. And then there's a third class that I like to class, and that is a problem that is associated with any genome that has a linear chromosome. By definition, it has an end. And that end could be seen, for example, as a double-strand break. And so what has happened is that the way that cells overcome this problem is they have evolved a complex structure called the telomere, which has a number of roles, one of which is to mask this end from being seen as a double-strand break. And of course, if you don't do that, all of the chromosomes fuse together. So why did we study this? Well, this has strong links to human disease, particularly to cancer. And these are just some of the examples of the syndromes that can arise as a result of problems with any one of these problems. Ataxia telangiectasia, Nijmegen breakage syndrome, which John actually has made a huge contribution to, is very much involved in the detection of double-strand breaks. Bloom syndrome and Fanconi anemia I'll talk a little bit about later on in the talk. Severe combined immunodeficiency, a problem in repairing double-strand breaks during VDJ recombination, and so on and so forth. So, and I should say this is by no means a complete list. This is just some of the examples. So the major pathway that we've been working on in the last 10 years in my lab is homologous recombination. This essentially is a largely accurate method of double-strand break repair. So for example, if you get a double-strand break in one sister chromatid or homologous recombination, the way you initiate recombination is to trim back the ends in a particular way that you have a 3' overhang. And this is a substrate for one of the major recombination proteins in the cell, which is Rad51. And this is just an electron micrograph of a filament of Rad51 bound to single-stranded DNA. And there's some beautiful structural work that's been done here at Sloan Kettering from Nikola Pavletic. This isn't actually his structure, this is from Phoebe Rice, but nevertheless we have a real molecular insight into how this process is working. So Rad51 loads onto here. It then has this remarkable ability to seek out homology, and this is what's happened here. It invades one of the broken ends into the intact homologous or sister chromatid, displacing the non-complementary strand. And this is called a displacement loop or a D-loop intermediate. And what this does essentially is to establish a replication fork, which can then be primed for DNA synthesis. This then fills in the gap, and using lots of fancy gymnastics, you can get repair events that basically completely repair or restore the broken molecule. So that's how it works. So this is absolutely essential in mitotic cells and in meiosis in eukaryotes, and has very strong cancer connections. Two of the most famous examples are the BRCA1 and BRCA2 breast and ovarian cancer tumor suppressor genes. So work over a number of years from my lab and many others has implicated BRCA1 in double-strand break resection, actually controlling the resection step in this process. And BRCA2, another of these very important tumor suppressor genes, actually plays a very important role in bringing the RAD51 protein to the site of double-strand break and then helping it to promote the strand invasion step of the reaction. So you can clearly see that there's a very strong cancer link here. John also mentioned Bloom's and Bloom syndrome, and this is another example I wanted to give you in relation to how recombination is controlled or constrained or actually channeled to different outcomes. So I've just added a little bit an additional complexity to this little schematic. So here what Bloom's does is basically to move these, this double holidase junction together to form a hemicatenane that's then disentangled by the action of topoisomerases. So Bloom syndrome basically results from a defect in the Bloom's gene, so an inability to promote this holiday junction resolution. And what happens is that repair now still happens, but it occurs in a way that's highly detrimental. So what we now start to get is crossover repair products, which the flanking markers are now exchanged. And the consequences of this are really quite striking. Bloom's is arguably the most cancer-prone syndrome that is known in humans. It's a broad-spectrum cancer predisposition. And the underlying reason for this is what some of you at the front can see, probably not at the back, but what's happened is that you've got crossover events, exchange events that occur within the sister chromatids, between sister chromatids. And what this does is it leads to loss of heterozygosity. So recessive point mutations in tumor suppressor genes are normally fine, but in the case of Bloom syndromes, these become homozygous and then they become a problem, which is almost certainly why this is such a horrendous cancer predisposition syndrome. So we've been very much interested in constraining recombination. We did a number of screens, firstly in C. elegans, and we've then extrapolated that to mouse models and human cells to try and identify factors that can control recombination in different ways. And several years ago, we described this helicase that John mentioned very briefly. And we did this initially in worms and then showed this is true in humans, that when you knock this particular gene out, recombination becomes out of control. So you become hyper-recombinogenic, and the cells become very sensitive to DNA damaging agents. And what we did was to actually show biochemically how this works. So remember, Bloom syndrome works much further down in the pathway. Where RTEL works is actually at this point where the RAD51 protein has generated this strand invasion intermediate called the displacement loop or D-loop. And what we were able to show is that RTEL has very specific activity towards this. And this is shown in a little bit of data here where we have formed a D-loop intermediate in vitro with purified proteins, RAD51, the strand exchange protein. And you can see that when you add increasing concentrations of the wild-type protein, this product disappears. So actually what's happening in this reaction is that this displacement loop is being reversed. So this really told us one way that RTEL can work is to reverse the recombination reaction, basically displacing the invaded strand to reverse the process. So what we've gone on to in subsequent work is to try and understand how this activity is used in different cellular contexts. So we showed in an original paper, the first paper that described this, that this activity is very important for counteracting toxic repair events, so when recombination has occurred in the wrong time or at the wrong place. But it turned out that this activity is also used in meiosis. So it turns out that when you knock out RTEL, for example, in the worm, recombination, specifically crossover recombination, goes up, suggesting that the activity of RTEL is important for suppressing crossovers through a role in non-crossover control. This activity is also used, at least in part, to help with telomere maintenance, and I'm going to give you a little bit of an insight into that with a few slides. So just briefly to introduce telomeres, as I mentioned, these are these specialized structures that exist at the end of all linear chromosomes. They're made up of the repeated sequence. This is the repeat for humans, TTA, GGG, and you can see that there's a single strand, 3' overhang, and a double-stranded telomere repeat sequence, and these can be 10 to 20 kb in humans. So what these specialized structures do is that they're recognized by a number of proteins. The sheltering complex, which was described very elegantly by Tisha de Lange's lab, and she's done amazing work on this complex. There are two double-strand telomere binding proteins, TRF1, TRF2, and POT1, which binds to the 3' end. And what these do essentially is to protect the chromosome end. They hide the double-strand break from being seen as a substrate for non-homologous end joining, for example. They also recruit and regulate telomerase in conjunction with the CST complex. And what this does is it solves the end replication problem. So normal semi-conservative replication can't complete replication to the very end, so you need telomerase to sort the problem out. And more recently, there's emerging evidence that actually the sheltering complex, specifically TRF1, helps to facilitate replication through this structure. This has the propensity to form secondary structures that can impact on replication. So this is certainly one area that's new to the field and is exciting. So this RTEL helicase I mentioned was actually first discovered in mouse as a regulator of telomere length, which is where it gets its name, RTEL. And the knockout mice for these are embryonic lethal, and when you take cells from these, there are really quite profound genome instability phenotypes, chromatid breaks, and telomere abnormalities. And this is just an example of what you see. So normally you would have two intact telomeres, and the way we're visualizing these is using telomere repeat fish. And what you can see is that when you inactivate RTEL, there's some really quite rapid and dramatic changes that happen to the telomere. The telomere gets shorter, and you can see that the spot intensity is significantly reduced, but they also get longer, and that the intensity of the telomere spot is increased. So this is proportional to the length of the telomere. There are also extreme cases in about 20% of chromosomes in any one cell completely lose detectable telomere signals. And we also see this really bizarre phenotype, which is multiple spatially distinct telomere fish signals, which is telomere fragility, and this is linked to replication. So what's this got to do with recombination? Well, this was the first visualization electron micrograph of a human telomere from Jack Griffiths and Tisha de Lange nearly 15 years ago now. And what this is a lasso structure, and it basically transpires that the telomere can adopt a specific configuration where the 3' end of the telomere invades to form this T-loop intermediate, telomere loop. And for those of you who've been paying attention, what this is, of course, is a D-loop. So this is a recombination intermediate that looks very much like a substrate for the RTEL1 helicase. So is this phenotype that I showed you where telomere loss and length changes somehow a reflection of a problem in recombination at the telomere? So biochemically, we've shown that RTEL can act on this substrate, a D-loop, to reverse or counteract the recombination reaction. So is the RTEL telomere phenotype a reflection of a problem in dismantling the T-loop structure? And that was really the premise that we set out to test a few years ago. And what we found, and I'll just summarize this in a model, is that during S phase, we believe that the approaching replosome triggers the recruitment of the RTEL helicase to the telomere, and using its D-loop unwinding activity, it can dismantle this structure. And this has two potential roles. It allows the replosome to complete semi-conservative replication, and also allows telomerase access to the 3' end to solve the end replication problem. So what happens in the absence of RTEL? Well, what we showed is that when you are RTEL deficient, we believe that the collision of the replosome potentially triggers the inappropriate resolution of this structure. And actually, we showed that this is actually dependent on a holiday junction resolvase called the SLX14 complex. So this is actually resident at the telomere during the cell cycle. And what we showed is that this actually cuts off the persistent T-loop, resulting in telomere circles. And this helped us to explain telomere shortening, loss, and also lengthening. So as John mentioned, our work is very much into the mechanistic aspects of RTEL, but in the last 6 months or so, there's been a large number of papers that have come out describing point mutations that are in the RTEL gene that are responsible for Euler-Horidason syndrome, which is a severe form of dyskeratosis congenita, which is a cancer predisposition syndrome. Furthermore, there's emerging evidence that mutations in RTEL, and interestingly also telomerase, or variants of them, are associated with a number of brain cancers, high-grade glioma, and also astrocytoma, and the importance of this in relation to what I've just told you about in terms of the telomere and recombination control is something that my lab is actively working on. So finally, I'll just tell you a little bit about other aspects of our work. So I mentioned that double-strand breaks are very important during DNA replication. And we've been very much interested in how interstrand crosslinks are repaired. And many of you will know that crosslinks are used as a first-line treatment. Cross-linking agents are used in first-line treatment for testicular cancer, for example. And what these do is they act as an impediment to both replication and transcription. So during the processing of these intermediates, you invoke the use of homologous recombination and also a disease a pathway called the Fanconi anemia pathway. So a number of years ago, again, starting in C. elegans, using it as a model, a genetic screening tool, we identified mutations in the HELQ gene that give rise to exquisite sensitivity to cross-linking agents. This was particularly interesting because about 30 years before our study, several groups identified mutations in Drosophila in this particular gene that confer sensitivity. So in the last few years, we've begun to try and investigate how this is, if anything, linked to human disease and what this gene actually does during the repair of damaged replication forks. So we made a knockout mouse, and we also did proteomics and biochemistry. And really the idea here is to see who HELQ actually works with, and that might give us some insight into what it's actually doing in the cell. To cut a long story short, what we found is that when we knock this gene out in the mice, you see subfertility, hypergonadism, cancer predisposition, sensitivity to cross-linking agents, and radial chromosome formation. And I'm sure many of the clinicians in the audience will realize what this is. It is Fanconi anemia, at least in the mouse. So this is, of course, a very rare, complex, multigenic disorder. The hallmark and also is used in diagnosis of the disease is the sensitivity of these cells to cross-linking agents. And these are manifestations of the problem in Fanconi cells, which are radial or quadraradial structures, which are the hallmark of this disease. So the HELQ deficient mice actually phenotypically were indistinguishable pretty much from other Fanconi mouse models, for example, FANCD2. And this was particularly interesting when we then did the proteomic analysis to find out who HELQ actually bound to. We pulled out three major complexes, ATR, A-TRIP, which actually recognize stalled and damaged replication forks. The RAV51 paralog BCDX2 complex, the RAV51 I've introduced to you, these are relatives of RAV51 that act at replication forks, and FANCD2 FANCI. So what does this mean? Well, it turns out that three of the genes that we found interacting with HELQ are actually mutated in Fanconi anemia, RAV51C, FANCD2, and FANCI, and two of those genes were also implicated in ovarian cancer susceptibility. And it was this particular interactions that got our attention, because it transpired that the mice that we had generated, the HELQ deficient mice, while having a late onset, these mice were particularly prone to ovarian and to pituitary cancers, with mice showing about 40% ovarian and 30% tumors within our cohort. So this was really exciting, and of course the resemblance to Fanconi anemia has now prompted us to look in Fanconi anemia-like disorders, those ones that are currently non-defined in terms of the genetic component of the disease, to see whether HELQ is a Fanconi gene or a Fanconi-like gene. What I didn't mention is actually the HELQ heterozygous mice, so losing just one copy of the HELQ gene is sufficient to give you the phenotypes that I have told you about, albeit at a slightly reduced level. And this is particularly interesting in line with the fact that HELQ is subject to hemizygous, so single copy loss in human serous ovarian cancer. Now, what we're now looking at is whether this is actually a causal event, a driver or a passenger mutation event. But nevertheless, I think when you combine this with the fact that the mice are haploinsufficient, I think this could be very interesting to pursue. And finally, and I didn't go into the details, we were able to show that HELQ actually works in the recombination branch of cross-link repair. So this obviously gives me an opportunity to thank a number of people who have funded our work. I'm particularly grateful to Cancer Research UK that support the institute that we work at. These are the current and past members of my lab over the last 10 years, and they've been a fabulous group of people, and it's been a real pleasure working with them. And this prize is very much a reflection of the work that they've done with me. And also the long list of collaborators that we've had. And I think John kind of pointed to this. I get the most out of science from talking to people and working with people, and it's down to this group of people, collaborators in Europe, US, Japan, all over the world, that make science so much fun. So thank you very much, and again, it's a great privilege to receive this award, and thank you very much.